Hello children, welcome back to the class. Today in this class we will discuss about the cell division. पिछले वीडियो में हमने डिस्कस किया था about the chromosome and its structure and its chemical composition. आज इस वीडियो में हम डिस्कस करेंगे the two vital process of the living organism, that is cell division, mitosis and meiosis. तो इस वीडियो को शुरू करने से पहले इट्स माय रिक्वेस्ट कि आप मेरे पिछले वीडियोस को ज़रूर देखें. इट विल बी ईजी फॉर यू टू गो थ्रू द चैप्टर. सो आई होप you will understand the video, you will like it, share it and subscribe it. So let's continue with the chapter. तो आज हम स्टार्ट करेंगे cell division, the most vital process of the living organism that takes place both in plant and animal cell. जो animal और plant cell दोनों में occur करता है. So there are generally two types of cell division. Mitosis and meiosis. Mitosis and meiosis. तो mitosis में क्या होता है? Here one cell, one parent cell divides into two daughter cells. Two daughter cells, and here the chromosome numbers does not change. That means chromosome का जो number है वो शुरू से लेके the entire process it remains same throughout the whole cell division. And in meiosis that generally takes place in the gametes. It takes place in the gametes, that is the sperms and the ova, and here the chromosome numbers becomes half. Because when the fusion of gametes takes place, again the chromosome number gets back to its original number. So this is the two main process of cell division, mitosis and meiosis. Mitosis, one parent cells give rise to or breaks into two daughter cells. Chromosomes number remains same throughout the process. And in the second phase, meiosis generally takes place in the gametes, that is the egg and the sperms. And here the chromosome numbers become half, and at the last stage again the chromosome they fuse and the chromosome number it retains back to its original number. So now we will discuss about another two important process that takes place during cell division is karyokinesis and cytokinesis. Karyokinesis is all the nuclear division, all the nuclear changes that takes place during cell division is called karyokinesis. All the nuclear changes takes place here. That takes place during cell division. And cytokinesis, that is the splitting up of cytoplasm. Splitting of cytoplasm that takes place at the end of the process. So here one more phase, karyokinesis and cytokinesis. In karyokinesis, the nuclear changes takes place during the cell division, and cytokinesis, all the splitting of cytoplasm that takes place at the end phase or at the end of the process. Now, karyokinesis has four main phases. Now, what are those phases? Prophase, metaphase, anaphase and telophase. So the process of karyokinesis, that is the nuclear changes that takes place in the cell, it has four main phases to go through. First one is prophase, metaphase, anaphase and telophase. So I think up to here students have understood. बच्चों आपको समझ में आया है कि सेल डिविजन क्या है, दो प्रोसेस क्या है, माइटोसिस और मेयोसिस. उसमें माइटोसिस में दो प्रोसेस होता है, कार्योकाइनेसिस और साइटोकाइनेसिस. एंड द कार्योकाइनेसिस जो एक न्यूक्लियर चेंजेस इन द न्यूक्लियस, जो चेंजेस आते हैं न्यूक्लियस में, उसमें फोर फेजेस है, प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. So अब एक एक करके steps. Now one by one we will study the steps with the comparative study with the animal cell. Animal and plant cell दोनों में क्या changes आते हैं that we will see through our diagrams. और सेल डिविजन में आने से पहले द सेल रिमेन्स इन अ रेस्टिंग स्टेज, जो हम लोग पिछले क्लास में डिस्कस कर चुके हैं वीडियो में कि इंटरफेस क्या है. इट्स अ रेस्टिंग फेज ऑफ द सेल ऑफ द न्यूक्लियस वेयर नो चेंजेस टेक्स प्लेस. The chromosomes just appears like chromatin fibers, nucleolus is present, nuclear membrane is present. So that is the interface जहाँ पे कोई change नहीं होता है. In animal cell as well in the plant cell, this is the interface. The resting phase हम लोग जिसको बोलते हैं cell division शुरू होने से पहले. Here there is a nucleolus is present, the chromatin fibers are present just like the network, and presence of cell membrane. Okay, and in plant cell you'll find again a nucleolus, the chromatin fibers just around like a network. We have the cell wall that's made up of cellulose, we have the cell membrane, we have the nucleus, the nucleolus, and the chromatin fibers. So this is the resting phase or the interphase. That is just before the beginning of the cell division, just before. Now we'll move on to prophase, the first phase, early prophase and the late prophase. We'll draw the diagrams and see how it occurs, what are the changes that takes place in the cell, in the nucleus of the cell. This is the animal cell. There is always a cell wall in the animal cell. There is a nucleus. Now, and this is the late prophase. बच्चों जस्ट रफ डायग्राम है समझने के लिए, अगर डायग्राम्स हम लोग पढ़ेंगे तो जल्दी याद हो जाता है. It get memorized quickly. So this is the animal cell, this is a plant cell, this is the early prophase, early. And this is the late. अब क्या changes है? Just look. The chromosomes become more distinct and visible. जो network form में था, अब देखो ये ज़्यादा concentrated हो गया एक जगह पे और एकदम साफ दिख रहा है. Now they are not arranged like a not like a network, but they have just concentrated. They have become more visible, more accurate. And in animal cell, one more change takes place. That is centrosome that is present only in the animal cell. It gets splits into two centrioles. जो centrosome है ये centrioles में split कर जाता है. Daughter centrioles जिसको हम लोग बोलते हैं, जो plant cell में absent है. In the later prophase, this chromosomes become more distinct. ये chromosomes और distinct हो जाते हैं और properly visible हो जाते हैं. And the centrioles they start moving towards the opposite poles. और जो centrioles है, दो centrioles वो opposite poles में move करना start कर देते हैं और धीरे धीरे एक connection centrioles और chromosome के बीच में, और ये जो nuclear membrane है वो धीरे धीरे dissolve करना start कर देता है. And the formation of spindle fibers takes place. Spindle fibers that connects just like a connection between the centrioles and the chromosomes. Chromosomes धीरे धीरे equatorial line, एक तरह के line में अपने आप को arrange करना start कर देता है. प्लांट सेल में सिर्फ centrioles are absent, spindle fibers बिल्कुल वैसे ही बनते हैं, and they started moving towards the opposite pole. This is the animal, this is the plant cell. Okay, so what changes takes place in the later prophase? The centrioles, the daughter centrioles, वो poles के तरफ move करना start कर देते हैं. Chromosomes that has become more distinct, they started arranging near the equatorial plane, and the formation of spindle fibers takes place. And asters can be seen. Asters are star-like structure. ये asters क्या है? These are stars-like structure, cellular structure, consisting centromeres and associated with the microtubules. ये जो हम लोग रे की तरह देख रहे हैं, microtubules during the early stages of the mitosis. ये नजर आता है, ठीक है? So this changes takes place in the prophase, the early and the later. Now we move on to the next step, that is the metaphase. अब धीरे धीरे क्या है, न्यूक्लियर मेम्ब्रेन, न्यूक्लियोलस सब डिसअपियर हो जाता है. प्रोफेज में जैसे हम लोग देखते हैं कि क्रोमोसोम्स बिकम मोर विजिबल, क्लियर, डिस्टिंक्ट. सेंट्रोसोम्स गेट डिवाइडेड इनटू टू डॉटर सेंट्रियोल्स, सॉरी, एंड द न्यूक्लियोलस, द न्यूक्लियर मेम्ब्रेन ऑल डिसअपियर्स. क्या रहता है वहाँ पे? एस्टल लाइक स्ट्रक्चर्स रहते हैं, स्पिंडल फाइबर्स रहते हैं इन दिस स्ट्रक्चर. और प्लांट सेल में क्या है? सेंट्रोसोम और सेंट्रियोल्स आर एब्सेंट. So directly only the spindle fibers are there. Now in the metaphase, each chromosome gets attached to spindle by its centromere. Now this is the animal, and this is the plant cell. Just look, जो क्रोमोसोम्स थे वो एकदम इक्वेटोरियल प्लेन में अपने आप को अरेंज कर लेते हैं. एंड हियर द सेंट्रियोल्स, they are distinctly moving towards the pole के तरफ मूव करते हैं, and they are connected with the spindle fibers. हर क्रोमोसोम स्पिंडल फाइबर से कनेक्टेड है. दिस इज द एनिमल एंड दिस इज द प्लांट. प्लांट सेल में सिर्फ हम लोग देखेंगे कि यहाँ पे सेंट्रियोल्स नहीं है, बस. Here are the chromosomes. And here are the spindle fibers that remains attached to the chromosome. Just look. Okay, so this takes place in metaphase. So distinct chromosomes hai, they slowly they are getting arranged towards the equatorial plane. At the center, centrioles gets divided, moving opposite towards the opposite poles. Formation of spindle fiber has taken place. Same thing is happening in the plant cell except the centrioles. Centrioles छोड़ के बाकी सारे changes same है. Now this is the metaphase. Now we'll move on to anaphase, the early anaphase and the late anaphase. क्या होता है? Different structures that are present. How it is? Early, the anaphase. This is the rough diagram. Now this is the animal cell, this is the plant cell. Now see in the early, what happened? The chromosomes, now they are splitting into two parts and moving towards the each centriole. See, centromere attaching the two chromatids divides. Now clearly the division is seen. It is more appropriate. Two sister chromatids of each chromosome separates. Now the sister chromatids that are present, one chromosome having two sisters is made up of two sister chromatids. Now they separate and they move towards the opposite poles attached to their respective centromeres. Pull by. Now by shortening of the spindle fibers takes place. And in the later and the sister chromatids, they start moving towards the opposite poles. They are attached with the spindle fibers, they become more shortened and thickened, and they start moving towards the poles, opposite poles. And in the late anaphase, formation of furrow take place. That means now the cell is going to be ready for the splitting up. Look, the centrioles are there. How the chromosomes now just move towards the pole, and now they become, see, plants as also, they are moving just apart. They are moving towards the pole apart from each other. This is. Now, so the sister chromatids, they move apart from each other, joining with the centrioles and moving towards the opposite poles, and the spindle fibers become short, thicken. Okay, now distinctly. And the later anaphase, we find that the formation of furrow takes place. That means now it's getting ready for the splitting up of the cells. The cells are going to divide into two daughter cells, having each of them are going to have one centrioles, and the formation of nucleus will take place in the next phase, that is the telophase. Telophase. Now in telophase, the furrow formation becomes more appropriate in animal cell. Now just look here, the centrioles are present on the opposite side of the poles, and here the nuclear membrane again reappears, and the chromosomes again becomes or changes to a thin chromatin fibers get arranged in the nucleolus, and the nucleolus again reappears. Cleavage furrow starts deepening. Now this furrow again deepens, and in the plant cell, formation of cell plate take place. First, so again the nuclear membrane reappears, nucleolus reappears, the chromatids or the chromosome that has formed, that has thickened, or those which has become concentrated, visible, now they're again converted into chromatin fibers, just like network of chromosomes. So this is the change in the telophase in the animal cell and the plant cell. And now at last the cytokinesis takes place, the splitting up of cytoplasm, and it gives rise to two independent daughter nucleus, having two independent cells having an independent nucleus. Chromatin fibers are arranged in the nucleolus. There is a presence of nucleolus and a centrosome. Each of the cell. This is the daughter cells. Okay, and the plant cell plate appears. Cell plates appear that divides the cell into two daughter cells. The parent cells get splits into two daughter cells, each having a nucleus with a nucleolus and the chromatin fibers. And in the plant cell, we also have the chloroplast and the mitochondria. They get arranged. They all reappears. All contents of the cytoplasm, including the mitochondria and chloroplast, randomly get distributed in the daughter cells during chirokinesis in plant cells, because chloroplast is not present in the animal cells. So here we come to the end of the mitosis. So children, what we have gone through, हमने अब तक क्या-क्या पढ़ा? We have studied about the mitosis, about the karyokinesis, cytokinesis, and the four phases of karyokinesis, that is prophase, metaphase, anaphase, and telophase. Now, what are the changes, the nuclear changes that takes place? That is in the structure of chromosome. In the first phase, we find that the chromosomes become thick, they are visible, they become distinct. The nuclear membrane disappears, nucleolus disappears, and the centrosome gets divided into two daughter centrioles in the animal cell. And in plant cell, centrosome is absent, so only the chromosomes that become distinct, and the nucleolus and the nuclear membrane disappears. Now after them in metaphase, they get arranged properly. They get attached to the centromere with formation of spindle fiber takes place, and in the telophase they get arranged in the equatorial plane, and starting of furrow. The furrow formation starts taking place. And the last telophase, the furrow formation gets completed and it splits into two daughter nuclei or two daughter cells. Cytokinesis, that is splitting of cytoplasm, that plays in the last phase, that is the telophase. When the formation takes place, and in plant cell, we have the cell plate that separates helps in the formation of two daughter cells. Now we will read the significance or the importance of the. Achha, before we go to the importance, we'll study about the difference between the animal and plant mitosis. In animal cell, asters are formed. Asters are star-like cellular structure that looks like star. Okay, these are asters like this. And so these are found only in animal cell because they are formed from the centrioles, whereas plant cell does not have asters. Cytokinesis by furrowing process, and here cell plates are formed in plant cell. In animal cells, it occurs in most tissues throughout the body for growth and replacement. And the mitosis in plants, it grows at the growing tips for lengthening and for growing in the breadth. Now we move on to the importance of mitosis. Why it is important? It is important for the growth of the body, repair of the damaged cell, replacement, and for asexual reproduction in the unicellular organisms like amoeba, like yeast. How they reproduce? They reproduce by division of the cell. That is from one parent to two daughter cells. In this way they divide and form a new and that daughter cell only, they again grow into an independent amoeba or independent yeast. So here for asexual reproduction, there is a requirement of the mitosis. Now maintain same chromosome, and one more important thing is the maintenance of the chromosome number, which remains same throughout the process. Now we will just discuss about meiosis little bit because today we will have only the discussion about the meiosis. As a meiosis is a process that takes place in the reproductive cells. Meiosis occurs in the reproductive cells. Generally takes place in the reproductive cells. Gametes, male and female. Sperms, ova. So the chromosome numbers becomes half. Suppose in human we have 46 chromosomes. Chromosomes in humans. Okay, so when the division starts, it becomes 23 in one cell, suppose the male, and again 23 in the female. Now this process starts. The chromosome numbers in each of the cell starts. It becomes half. But ultimately, when there is a fusion takes place of the gametes, again after the fusion, the number becomes 46. So here again the number gets back to it retains the original number 46. Okay, so this generally the main change that takes place in the process of meiosis. Now what is the importance of this process, meiosis? Chromosome number is halved in gametes so that on fertilization or in fusion it again restored, the number is again can be restored. The maternal and paternal chromosomes get mixed up during the first reduction process as a separate from the homologous parent. We have already seen in the mitosis how the sister chromatids splits. They splits and move into different poles. So here also when splitting takes place, what happens? The parent chromosome and the maternal and the paternal chromosomes, they stick together. And that is they come together and crossing takes place. That is, crossing of characters takes place. While the maternal and paternal chromosomes are separating, the chromatid material very often exchange between the two members of a homologous pair. So the parental chromatid, the maternal paternal, when they comes together, crossing of the characters also takes place. This is known as crossing over, and it results in genetic combination, recombination. Then chiasma. Chiasma, that is the egg shape. The chromosomes gives a egg shape. This formation is called chiasmata. This egg-shaped structure formed due to crossing over between the non-sister chromatids of the pair of homologous chromosomes. These are the non-sister, that means not belonging to the same cell, one from the paternal, one from the maternal. Maternal and paternal. Non-sister chromatids. This is called crossing over, leads to the formation of chiasma. So this is the significance, and this is the process what takes place in the meiosis. Rest of the process is just at the same as the meiosis that we have discussed. Only when the sister chromatids, when the chromosomes, when they split into two sister chromatids and they move towards the poles, at that time during the fusion, the maternal and paternal chromosome, they comes together, and this is called crossing over. And crossing over results in the recombination. That results in an egg-shaped structure called the chiasma or chiasmata. This is egg-shaped structure of non-sister chromatids. So I hope children, today in this class you have understood the cell division process, mitosis and meiosis. It's important and its importance. And so if any problem, any questions you have, you please put it in the comment section. और जिन लोगों को समझ में नहीं है, I request them to just follow the previous video. उस वीडियो से इस वीडियो को देखेंगे तो आपको कंटिन्यूसली समझ में आएगा how we have continued the chapter and we have ended here. So I hope you have understood the class. If any questions you have, any queries you have, just put it in the comment section. So thank you, and I hope you will like it, share it and subscribe it. Thank you.